Hi, my name is Alba Mayra Padilla. I'm a medical student in Universidad de Anáhuac Querétaro, and I'm doing a research internship in the Laboratorio Internacional de Investigación sobre el Genoma Humano with Alejandra Medina and Ana Beatriz Villaseñor. In this study, we want to know the gene regulation of Hedgehog interacting protein, HHIP, in chronic obstructive pulmonary disease. COPD is a progressive, irreversible, and disabling disorder that causes respiratory insufficiency. And it is estimated by the World Health Organization that COPD will become the third cause of mortality in 2030. Clinical manifestations and genomic profiles are heterogeneous and change across individuals. In past studies, we found that Hedgehog interacting protein is overexpressed in COPD patients. HHIP works as a negative regulator of the Hedgehog pathway and also has been shown numerous polymorphisms associated with COPD. Therefore, our objective is to understand if COPD-associated variants in the regulatory region of HHIP affect the motif binding sites of transcription factor that regulates HHIP expression. I would like to highlight two main steps that we used in this study. First, using PulmonDB, there is a web database of gene expression. We extract COPD experiments that were done in lung tissue. Then, using PySenic, a tool where we can see regulatory networks, we calculated co-expression genes that may be co-regulated by one transcription factor. Using the banding motifs and selected SNPs associated with COPD, extracted by manual curation, and different public databases like GWAS, ClinVar, GTex, and RegulomDB. We use variation scan. There is a tool that was developed in our lab, and it estimates the effect of an SNP variant in the transcription factor motifs. As a result, in figure number 1a, we can see the HHIP expression in three groups: controls, COPD, and IPF. We used some samples from idiopathic pulmonary fibrosis, IPF, because it's considered an antagonism, a chronic disease of COPD, and both results in lungs scarring. We found a statistical significance among groups, and the change between the control and COPD group is smaller than COPD versus IPF. In figure 1b is a heat map with the gene expression across all components of the canonical Hedgehog pathway. Columns were clustered and 5 groups were considered to have biological significance. One with COPD and IPF samples, another with controls, then a group with substantially more COPD than controls, another with IPF samples, and finally a cluster with a mix of COPD and controls. In figure 2, using all genes shared in the microarrays, around 14,500, we perform an analysis using PySenic. The results show 12 transcription factors that regulate 293 regulons, which include HHIP. An example of one regulon can be seen in figure 2b, in which the transcription factor LHF, that means hepatic leukemia factor, regulates 40 co-expressed genes, including HHIP and SHH member of the Hedgehog pathway. In the human genome, HHIP is located in chromosome 4, and in figure 3, we are shown HHIP and its regulatory region. You can see SNPs that we search in scientific literature by manual curation in purple lines, and SNPs from different databases in green lines. Then, we use variation scan with the SNPs to know if some of these variants affect the motif of the transcription factor that regulates HIP, and it gave us 3 results. And there is one that can be very interesting, variant RS10322295. We can see the LHF motif and the different alleles in color. The risk allele for COPD is the major, and it is indicated with a green star. It means that COPD patients are associated with the allele A. When we have this allele, the transcription factor LHF have more affinity and it leads to have HIP overexpressed. Moreover, with the minor allele, LHF lose the affinity to the DNA. As a conclusion, there are variants associated with COPD that can play a regulatory role and have an impact in the motifs of the transcription factor. And we found that the risk allele for COPD is the A, and it has more affinity for the LHF transcription factor subserve HIP expression. Finally, I would like to thank all my lab mates and collaborators, and I will be happy to take any further questions.